Well, good morning, everybody. Thank you so much, Sandy, for the introduction and for putting me first on a Saturday morning after a big banquet. That's really, it's got to be the worst location in the agenda. But so congratulations to everybody for staying awake and getting up early. I'm going to try and review with you in the next few minutes some of the complexities of clinical trials. And what I'd like to start with is ask for a little audience participation, perhaps a show of hands, who, from a patient standpoint, has participated in clinical trials, who's been in a clinical trial. So we've got a few people. And from a healthcare provider, either physician or other healthcare provider, who has been involved in running or organizing a clinical trial. Great. So we've got a few people on each side of the spectrum. And what I wanted to do today was review with you and do a little bit of Clinical Trials 101. And we sort of take for granted that we're all on the same page when we talk about a phase 1 trial and a phase 2 trial and informed consent and randomization. And I thought I'd take a step back and go through the process with you in the next few minutes. So this distinguished couple that you see here are the Laskers, who founded the Lasker Award, which is sort of a couple of notches below the Nobel Prize. And Mary Lasker, who's shown here, has a wonderful quote, which I wanted to share with you. If you think that research is expensive, try disease. And of course, this is really what drives a lot of what we're talking about here and have been talking about in the last few days, both from a scientific point of view and also from a clinical research point of view. Research is expensive, but in the long run, the societal effects can be very impressive. And I'll actually show you some data at the end that speak to that. Okay, so what about clinical trials, and why do we do clinical trials? And this is sort of a joke scientific article that was published this year on parachute use. And let me ask for another show of hands. Who in this room thinks that it is proven that parachutes save lives? And since we're all scientists, whether we're physician scientists or patient scientists, who'd be willing to participate in a randomized controlled trial of parachute use? Not many hands go up. So the point is, we take a lot of our scientific basis for clinical research on faith. Many things, many treatments, many diagnostic studies have really come into broad-scale use, not from clinical trials or an evidence-based approach, but more from an experience-based approach. Obviously, this clinical trial is never going to be done. So we use that phrase, evidence-based medicine, a lot. And in fact, what does it mean? So I thought I'd go through some of the stages or levels of evidence that go into an evidence-based approach to medicine. And increasingly, medical students now are being taught to rigorously look at the literature, analyze how studies have been done, whether they were done appropriately, correctly, and whether the inferences or the assumptions based from those studies actually were supported by the evidence in the study. And so when you go to, hopefully when you go to a doctor in the next 5 to 10 years, they'll often be much more savvy about the literature and be able to pull out articles and construct your treatment based on evidence rather than simply experience. Not that experience isn't important, obviously it is, but increasingly we're relying on an evidence-based approach. So let's go through the levels that lead to evidence-based medicine. Now, the first level, and if you will, the highest level in this hierarchy, is evidence from a meta-analysis or a combination of several randomized controlled trials. So if, for example, we wanted evidence that treatments for transverse myelitis were effective, we'd really like to get a series, an average of several randomized controlled trials. We don't have that for transverse myelitis or for multiple sclerosis for that mind, but we do for some other neurological diseases. Now, one step down, level 1B, is evidence from at least one randomized controlled trial. And again, here in multiple sclerosis, we have a number of randomized controlled trials that have led to treatments that have led to this evidence-based approach. One step further down is evidence from a well-designed control study without randomization. I'm going to talk a little bit about randomization later and explain what that phrase means. So randomization is where individuals are not randomized to one intervention compared to another intervention. One step further down, evidence from one well-designed quasi-experimental study. So that means that rather than being in a randomized, controlled clinical trial, an individual or a series of individuals, their experience with a drug or an intervention, a treatment of some kind, is collected in a systematic way, but they're not in a randomized control trial. And then further lower levels of evidence, if you will, are from descriptive studies, such as cohort studies or case studies, where a number of patients who have similar signs and symptoms, similar diseases, and have been treated in a different way are just collected together. And finally, the lowest level of evidence is really based on clinical experience. Going to a series of docs, for example, us, who've treated transverse myelitis for a number of years and saying, well, what do you think works? So again, not to belabor the point, but this is really a hierarchy. So as you look at clinical data from a clinical study or a clinical trial, you want to look and see what kind of evidence, what level of evidence comes into this. Is it a high level of evidence or a lower level of evidence? So let's go back a little bit and talk a little bit about the history of clinical trials. I'm very interested in medical history, and it's worth reviewing how we got to where we are now in terms of clinical trials. And it may surprise many of you that clinical trials predate Christ, and the first clinical trial, in fact, was conducted by King Nebuchadnezzar in 605 BC. And he ordered several of his children to go onto a strict diet of meat and wine, and several of the kids did not want to go onto this diet, and they wanted a more simpler diet. And at the end of about 10 days, King Nebuchadnezzar noted that the children who were on the simple diet, non-rich food, bread and water essentially, actually looked more healthy than the kids who were on the big fat, pork cutlets and red wine. So if you will, this might have been the first clinical trial, even though it wasn't conducted with a true design. What most of us think of as being the first clinical trial was conducted by this man here, Captain James Lind, who was a Royal Naval surgeon, and he did a true controlled clinical trial. It was based on the observation that sailors on long transatlantic voyages, if they were not given fresh lemons or limes, they developed a disease called scurvy, where their gums became rotten and bled easily, and their teeth became loose. They became very fatigued and listless. And sailors who were fed limes or lemons received vitamin C. This is the active element that prevents scurvy. And so he designed a controlled clinical trial where half of the sailors on the ship received rum, the other half of the sailors received regular rations of lemons and limes. And guess what happened? Of course, those who were fed the vitamin C enriched diet did not develop scurvy, and those who were fed the rum got scurvy and stinking drunk. So this, in fact, what most of us think was the beginnings of clinical trial research. In the 19th century, we began to see the emergence and acceptance of the use of placebos. Now, the word placebo comes from the Latin word meaning I please. I please. And the concept of a placebo is that it's a non-effective or an inert substance which is given perhaps to create a psychological effect, and we all know that the placebo effect can be very strong. But in the context of experimental research, a placebo is given to balance the active intervention that you wish to test. Now, into the 20th century, trials utilizing randomization began to occur. And randomization is basically a flip of the coin whereby if you have 10 patients who are in a clinical trial, you randomize or randomly assign 5 of the patients to one treatment, perhaps the active intervention, and 5 patients to the placebo, the inert substance, which is, if you will, the comparison group. And this is a process that often confuses patients because they think, well, why on earth would you want to do that? Why can't I choose which group to go into? And the process of randomization is really important for a high level of evidence-based clinical trials, because what it does is allow for the groups who are participating in that study to be balanced. Rather than Joe Smith saying, well, I have a belief that treatment A is going to be better than treatment B, and therefore I want to be in that group, we are able to balance subjects so that the trial is more homogeneous. And at the end of the trial, we have essentially very similar subjects or patients in treatment A group compared to treatment B. Now, the other thing that happened in terms of clinical research, which is an important milestone, is the development of the FDA. And the Food and Drug Administration, which we sort of take for granted now, evolved during the 1930s, 1940s. And for the first time in 1941, it was actually required to attest to the purity of a drug, and this was insulin. In the 1940s, we saw the development of multi-center studies. So most trials up till then, just like with Captain James Lind, had been done in a single center, a single academic center, or a single hospital. And now, with better communication, more rapid transportation, we were able to develop studies that involved multiple centers. So a university in California could be in the same trial using the same design as in Baltimore. And this had the major advantage of speeding up the process of clinical research. So you could rapidly enroll large numbers of subjects, all with the same protocol. Now, after World War II, one of the consequences of the Nuremberg process was the establishment of ethical points for the protection of subjects in trials, and this is called the Nuremberg Codex. And I'm not going to go through this in detail, but this was really the first document that laid out in detail the rights of individuals who participated in research. And up until that point, obviously most of you are probably aware of some of the atrocities that occurred in World War II that dealt with medical research on uninformed or non-consenting subjects. Now, we're into the 1960s now, and a couple of very sentinel events happened in the 1960s that further strengthened the responsibilities, the ethical responsibilities of clinical researchers to protect the safety and to develop in the true informed consent for subjects. So the first milestone was when Henry Beecher, a prominent anesthesiologist at Harvard, reviewed 22 separate trials and published in the New England Journal of Medicine a very scathing report about the unethical nature of those trials. So this opened a very broad public debate about the ethics of clinical research. Just as an example of what was going on at that time, and as you read back on these things, you sort of think, well, what were they thinking? And I'll just give you a couple of examples. So in one of the cases that he investigated and published on, live hepatitis virus was given to mentally retarded inmates or residents at a state institution in New York. And we think today, well, how on earth could people get away with it? But of course, at the time, there were no safeguards, there were no written documents to really guide ethical clinical research. Another one was where investigators in New York injected live cancer cells into patients with advanced dementia. They knew that these patients were quite likely to die from their dementia within the next year or two, and they wanted to look at the trafficking of these cancer cells. And again, today, this would never pass our ethical or institutional review boards, the boards that govern clinical research. But at the time, again, there were no guidelines to do this. Then finally, the last example that I want to give you is that of the Tuskegee experiment, and many of you have probably heard about this and may even have read this wonderful book called Bad Blood, the Tuskegee Syphilis Experiment. This was an experiment, or really a natural history study, that began in the 1930s and continued for 40 years in Tuskegee, Alabama. And what the investigators were trying to establish was what was the true natural history or natural course of syphilis. And in 1930, of course, we did not have penicillin. So the treatments which were available at the time for syphilis were pretty ineffective and quite toxic. So many of the 400 men who were enrolled into this study were not treated or were treated relatively ineffectively. During the course of the study, remember it was a 40-year-long study, penicillin became widely available and, of course, was used first in the military to treat individuals with syphilis. The tragedy of the Tuskegee experiment was that syphilis treatment, as it became more widespread in the general population, was not applied to men who were in the Tuskegee experiment. So essentially treatment was withheld. And this led, as this whole experiment and experience was made public, obviously led to a great deal of reform and, if you will, soul-searching on the part of clinical researchers. And this led to what we call the Belmont Report in 1979, which laid out, again, further principles for conducting clinical research ethically. So that's really the history and the background, and I think it's important to see how we've got to the point that we are. And when we, as investigators, have to wrestle with our informed consent process with our institutional review boards, we need to think back to where we were 30 to 40 years ago. In fact, it's not that long since Tuskegee experiment ended. So what are the purposes of clinical trials? So clinical trials are indeed human experiments. There are three fundamental principles that we need to think about with any clinical trial. It must address a legitimate question. You might have a scientific question that's of value but has no clinical relevance, and that probably does not have a place entering into a clinical trial. The second principle is that the patient must be informed and willing to participate. This is really the primary cornerstone of conducting clinical research in an ethical manner, that it's done with the patient and the physician or the clinician in a partnership, so there is true informed consent. And the third principle is that the patient may decline to enter the study, no matter how much subtle or sometimes not so subtle pressure is put on the patient, and can withdraw from the study at any stage. And there are four distinct clinical trial structures, and I'm going to talk a little bit about how phase 1 trials differ from phase 2 trials and from phase 3 trials. So first, a phase 1 study. This is, if you will, the first stage in the clinical testing of a new drug or a new intervention, and it provides very basic pharmacological and toxicity information. It does not tell us anything about therapeutic efficacy. So if you're looking at a clinical trial and it says a phase 1 trial, remember that that study was not designed to tell you whether that drug was effective. It was simply designed to give us pharmacological information. Is the drug absorbed? What kind of blood levels does one achieve? And is it safe? Does the patient taking the drug have a good response without adverse effects? This can be done in healthy volunteers. You don't have to have a neurologic disease or a systemic disease to participate in a phase 1 study. In fact, most drugs are first tested in healthy controls. You probably heard of this recent tragedies in England with healthy college students who received an investigational drug, and several of them ended up in the intensive care unit. These were healthy controls participated in a phase 1 study. But phase 1 studies can also be performed on patients whose disease has progressed. Now, I'm going to skip this, and since I talked about randomized clinical trials, I'll move on to talk about phase 2 studies. Okay. We sometimes have some confusion between phase 1 and phase 2 and phase 3. A phase 2 study is really used to identify the dose range of a particular drug. We've got a drug, we've shown in a phase 1 study that it is well tolerated. Now we want to find what dose is the best tolerated dose, and also to give us a hint of efficacy. These are usually relatively small studies, 20 to 50 patients, and these are usually done in people who have the disease that we're planning to test the drug in. So after a phase 2 study, we'll then move on to a randomized phase 3 trial. This is usually what we read about in the newspaper when there's a discovery with a new drug or a new procedure, intervention of some kind, and the newspaper talks about a new drug being developed. It's usually from the results of a phase 3 study. Phase 3 study compares the effects of different treatments, and this could be a placebo compared to the active treatment, or it could be the active treatment that you're trying to test against the standard treatment. For example, when we're testing 2 antibiotics, a new antibiotic against a conventional or standard antibiotic. The outcome measures in a phase 3 study usually involve the response to the drug. We look at toxicity. In certain diseases, for example, HIV, we look at survival. And phase 3 studies typically are large, so they have to be multi-center. They sometimes are even multinational. Now, after a phase 3 study, if the data is convincing enough, the pharmaceutical company can submit those data to the Food and Drug Administration for what we call registration. And in general, at least two phase 3 trials have to be performed before a drug is registered or approved for release for prescription on the market. I'm going to say a few words about trial design. And again, as you look at informed consent, there are 3 main types of clinical trial design that you as potential patients participating in a trial or healthcare providers organizing a trial will know about. This is the most simple. This is a basic randomized clinical trial design. So we have our population of patients. If this is a phase 1 study, as I mentioned, this could be healthy controls. These are the potential individuals who are going to participate in the study. And the first step is really the informed consent process, whereby the investigator or study coordinator will sit down with the potential subject, describe the study in detail, and give them the informed consent to read. This is often a very lengthy document. It can be 5 pages, 10 pages. It's supposed to be written in simple to understand language. And then the subject will actually be asked questions. It's kind of like a test. What did you understand about the study, and do you have questions about the study to see if they're comprehending the informed consent document. Now, if they sign the informed consent document, by definition, they're now in the study. And then there is usually a screening period where we do screening blood tests to make sure that it's safe to be in the study, or we do an MRI scan to make sure that the patient has the right number of lesions, if it's multiple sclerosis, or the right pattern of disease to be in the study. And after the screening process, randomization occurs. And here, it's literally done by a flip of the coin. Usually these days we don't use coins, we use a computer to randomize patients either to the active treatment or the placebo arm. Now, most studies are designed as blinded or masked studies so that neither the investigator nor the patient participating in the study know whether they're in the active treatment or the placebo arm. And of course, this is very easy when it's a tablet, because a placebo tablet can be made to look just like an active treatment tablet. It's less easy when a treatment medication has a very characteristic set of side effects. For example, we're working with a medication right now that has a very strong fishy odor. So all of the subjects in the study know that they're on the active drug because they have this fishy odor, and they don't like it, but it does tell them which arm of the study they're in. So that study, sort of by definition, unblinding has occurred. But most studies, we attempt, wherever possible, to maintain the blind so that neither investigators nor subjects know whether they're in these two treatments. So the trial proceeds. It could be several weeks, it could be several months, and then various outcomes are measured. In the context of multiple sclerosis or neurologic disease, there are often functional outcome measures, walking times, how quickly you can use your arms and hands, etc. Or it may be MRI outcomes. Now, the second type of trial, which is often used, is what's called a crossover randomized clinical trial design. Here we have our study sample. Randomization occurs just like it did in the parallel trial design. And here, individuals are randomized to begin the study, either to go on to treatment A or treatment B, and one of these might be placebo. And then after a few weeks, there is a switch. Individuals who were on treatment A will now switch across to receive treatment B, and individuals who were on treatment B will switch across to receive treatment A. This is what we mean by a crossover design. This is not used very often in context of multiple sclerosis or TM or most neurologic diseases, but it can be used for certain neurologic diseases, for example, headache or migraine. Now, briefly, there's a third type of randomized clinical trial design to test combinations of drugs, and I think you're all familiar with the concept from cancer chemotherapy that using several drugs is usually much better than using a single drug. Certainly that's what happens in HIV and in infectious disease. And in fact, in multiple sclerosis, there are several combined or combination trials now of disease-modifying therapies. And so in doing a factorial design, the main purpose here is to, after randomization, individuals may be on drug A and B, drug A and placebo, so they're only on one drug, placebo and drug B, or two placebos. So this group obviously is not actually receiving any active drug. And we analyze this in a factorial manner, comparing each of the four treatment-assigned groups. So how does all of this play out as we develop a new drug? So let's go through the timetable for a new drug development. And I think one of the most important things and most frustrating things for many patients is to realize that this, in general, is measured in years. So on average, it takes a new drug about 10 to 11 years to be developed from the time that it is tested first in the lab and then in animals, what we call preclinical testing, through our phase 1 study, where we're looking at safety and tolerability, phase 2, where we're getting a sense of what's the best dose, through phase 3, where we're getting a sense of the efficacy of that treatment compared to placebo or standardized treatment. The process at the FDA can take a long time. It can take several months to even several years for some drugs. On average, it's about 2 years. So even when you know that that phase 3 trial that you participated in has finished and closed and you're no longer going for study visits, a lot of work is still being done behind the scenes in Washington at the FDA to review the data from that trial, to compile it, and to decide, and this is usually done by an advisory panel, to decide whether a drug should be released. And when a drug is released, then it enters this, what we call phase 4 process, where we continue to do post-marketing surveillance looking for further side effects or further aspects of treatment. So I've talked a bit about informed consent, and I'm not going to belabor this, just to say that every clinical research study, whether it's a trial or an observational study or an imaging study, has to have informed consent. And these are very important documents, so please learn to read these very carefully. These should sum up for you the purpose of the study and what will be expected from you, and it will also define what the risks and benefits are from participating in the study. And benefits might include additional testing, for example, an MRI, or they might include financial benefits. So just like when you sign a mortgage on a house, read the small print. Now, increasingly, patient safety has become more and more of an issue, and most academic medical centers now are very highly focused on patient safety for several reasons. First is, in 2001, the Institute of Medicine published a report called "To Err is Human," and this detailed the fact that about 98,000 deaths occur each year in U.S. hospitals as part of clinical care through some kind of mistake, through some kind of what we call iatrogenic error. So iatrogenic simply means by doctors. 98,000, that's a tremendous number. And so most hospitals now have very active safety programs. And so too do we at Hopkins. And we also have a very active safety program within our clinical research arms. Part of this was triggered by the tragic death of Ellen Roche, who was a technician here at Hopkins, healthy control, participated in a clinical trial of an inhaled drug and died from that exposure. And her death and the death of other patients, either patients not in clinical trials or in clinical trials, has really led Hopkins and other institutions to make safety the #1 priority. So what do we do with all of these data after we've collected it? So I'll talk first about how not to compare treatments. Galen was a famous Roman physician just after the death of Christ, and he analyzed his response to patients who were given some kind of remedy, I don't know what it was, in this way. So all who drink of this remedy recover in a short time, except those who it does not help, who all die. Therefore, it's obvious that it fails only in incurable cases. And so what this really means is that this is one of these paradoxical statements, and obviously somebody who dies is not going to respond to the treatment. This is what his hypothesis was. But of course, there may be many other reasons why that patient died. Most noteworthy, of course, is that the treatment had no effect whatsoever on that patient. So we have to look at studies in a much more critical way than Galen was able to do. The other aspect of analysis is what we call intent to treat. And again, this is a concept that I wanted to explain to you because it's become a another of these cornerstones in analyzing clinical trials. And the cornerstone is really this, that all patients should be analyzed in a clinical trial in the arm of the study to which they were allocated at the time of randomization, irrespective of what happened to them. Now, this is a little bit different than what we do in clinical practice, where a patient is put on a drug, we look at their response to drug, we know that they're on the drug. Remember, in a clinical trial, we're often blinded to which treatment they're on. And so I wanted to give you an example. The best example I could find was actually from the cardiac literature. This is what we call the Coronary Drug Project that randomized patients to receive a cholesterol-lowering drug called clofibrate. And we had data in this study as to the adherence or the compliance of patients with their clofibrate treatment. And individuals who had poor compliance, by the end of the study, 25% of them had died. So you might say, well, poor adherence to treatment, the drug might work. If they're not taking the drug, then they're not going to have that response, and they're at a higher likelihood of dying. Individuals who had good adherence to medications only had a 15% mortality. So that looks like a big difference between those who took the drug and those who didn't take the drug. But in fact, the way that the trial was designed was to analyze all of the subjects in the trial and to use this intention to treat analysis, because we all know, for any of us who take medications, that it's really impossible, or pretty much impossible, to take every dose of a drug when it's prescribed. We all miss drugs and doses of drugs. So a more real-world situation is to do an intention to treat analysis. And this is to ignore adherence, so to combine everybody together, to compare clofibrate to placebo. And you can see here the differences become very much smaller. So 19% of the patients who got placebo died during the course of the study compared to 18% on clofibrate. So there's only a difference of 1%. In fact, these differences were not statistically significant. And this is the appropriate statistical analysis to use. And so you'll often read in clinical trial research whether it was an intention to treat analysis, and that's the appropriate way to analyze data. Now, another thing that's often done in the statistical analysis of clinical trials is to break subjects out by subgroups. Again, I have an example of this that shows, again, for a cardiac study, how doing a subgroup analysis can give you a statistically positive result that is not clinically meaningful. So in this study, patients either received aspirin or placebo, and the outcome that we were interested in was the number of deaths. And the investigators, I have no idea why, collected birth signs. So whether individuals were Pisces or Cancer or Libra, and they did a subgroup analysis based on those birth signs. And interestingly, as you can see here, individuals who, let's see if I can get this right, anybody who was a Gemini or a Libra had a higher death rate than all of the other birth signs put together. So, I mean, you can, if you're astrological in your viewpoint, you might consider this important. But what this really is used, or the point I'm really trying to make here, is that this is an example of a subgroup analysis where a clinically meaningless subgroup analysis is done. The study is broken down and analyzed really at a ridiculously small level or fine-grained level. So in general, be wary of subgroup analyses unless they're of clinically meaningful. Now, misconduct. Again, the newspapers have been full of statistical, scientific misconduct in the last few years. And I wanted to just pull out a couple of points so that you understand the difference between fraud and bias. And fraud, there have been very few examples of true scientific fraud. Most times, the problems in scientific studies result from bias that is not being handled properly. Bias might mean that an investigator is advertising a clinical trial and somehow oversells the potential impact of that treatment and somehow recruits individuals, and the individuals participating in that trial end up being imbalanced, meaning that they don't represent the general population of people with that disease. Now, unfortunately, you probably all have seen photographs like this. This is a true example of scientific fraud where a South Korean investigator had to apologize and retract all of his scientific observations on stem cells because of true scientific fraud. And as I've already said, this is fortunately a relatively rare event. Now, research fraud can work in both ways. Obviously, the picture I've just shown you is of a scientist manufacturing data or manipulating data in such a way. Sometimes patients are responsible for scientific fraud, and it can work in that way as well. So this is the age distribution for a study, and you'll notice that almost all of the patients in this sub-study are less than 40 years of age. Very few are over the age of 40. And the investigators actually went back at the end of the study because they were intrigued by this age distribution. It just looked a little bit odd. And it turned out, once they looked at the true dates of birth from Social Security numbers on these patients, that many of the patients had actually been constructive or creative about their ages. And there's a wonderful quote from Oscar Wilde that some of you may know. 39 is a very good age. I know many women in London who, of their own choice, have remained 39 for several years. So again, scientific fraud or the manipulation of data can occur not only by the investigators, but sometimes the patients don't give us exactly the right answer because they are very eager to be in a study. In fact, what happened in this study was the patients in this study knew that the investigators were looking for younger patients, and so there was an inherent bias to sort of shave a couple of years off your age. Data errors. I'm not going to go through this. Far and away, this is the most common and widespread problem in clinical trials. Much, much more common than scientific fraud, much, much more common than mistakes or errors that the patient may make. And this relates sometimes to the collection of data. And you're all familiar with the fact that up until the era of MRI, we used clinical scales very widely in multiple sclerosis trials. You've heard of the Expanded Disability Scale, the EDSS, and this was used to assess functional abilities in patients with multiple sclerosis. And recently, the National MS Society has developed the MS Functional Composite Scale, and this is, we think, a much better, more robust, more reproducible scale for clinical trials, which can be combined with imaging results to look at outcomes of treatment. I'm going to skip this just in the interest of time. So where do you get information about clinical trials? For multiple sclerosis, there are a number of very good websites, including the National Multiple Sclerosis Society. The NIH publishes a website called clinicaltrials.gov, and most reputable clinical trials that are listed on this clinical trials site. So finally, I just wanted to present to you just a brief synopsis of a recent paper, which I think is an extremely important paper, particularly as we go into an era when NIH funding is reduced. We now estimate that the NIH budget is about 7% less than it was in 2003 relative to inflation. And Clay Johnston at the University of California, San Francisco, published this study this year looking at the public impact of clinical trials. And he looked specifically at about 30 trials which had all been financed or funded by the National Institute for Neurological Disease and Stroke. And this shows the number of trials, there were about 30 over a 10-year period, financed by the Neurological Institute at NIH. And then he looked at different trials within this portfolio. Some of these are for multiple sclerosis, others are for stroke, for example, TPA, which is the clotbuster drug. And he did a very careful statistical analysis of the cost of the study and the outcomes of the study relative to outcomes on the health of the patient and the societal impact. So, for example, if a study had a positive result on stroke, getting people back to a functional lifestyle or to work would have a societal impact, and that could actually be measured in terms of dollars and cents. And amazingly, the portfolio here had a very positive result. Although these drugs, of course, have led to increased expenditure on healthcare because drugs cost money, the resultant health benefits had a much greater value than the costs, even when he evaluated that on a very conservative nature. And in fact, for those of you who have mutual funds or stocks and bonds, the yearly return on the investment from NIH through these clinical trials was 46% per year. So I think this, at last, is good ammunition that we have that clinical trials, certainly NIH clinical trials, do have not only a functional impact on the lives of people, but a financial impact on society as well. So I'm going to close there just to restate the principles of clinical trials. The 4 principles that I believe should be right up front and center in any clinical trial are to ask an important question, to answer it reliably and ethically, to publish the results, because if a clinical trial is done and completed and it never sees the light of day, you might just as well have not done the study. And it should be published whether it's negative or positive. And finally, the subjects who took part in the trial should be informed about it, and we should use those results to improve the next trial. So I'm going to close there, and I appreciate your attention, and thanks for the invitation to be here at this really a wonderful symposium. So thank you.